Hello guys, welcome back to my channel Botany Educator, and today we will be going to study about Rhizopus. It belongs to subdivision Zygomycetina. It includes two classes, Zygomycetes and Trichomycetes. Today we will study about Rhizopus, which belongs to class Zygomycetes. If we talk its journal characters, the members are saprophobes or weak parasite. Few are coprophilous, means it grew on dung. The plant body is well developed, filamentous and cenocytic type. Cell wall is made up of chitin and chitosan, means the members in the zygomycetes belongs to chitin and chitosan. In if we talk of earlier members, the cell wall was made up of fungal cellulose. So it's an upgraded cell wall in which the cell wall is made up of chitin and chitosan. And then asexual reproduction is through non-motile spore. In oomycetes, we have studied it's through motile spores, zoospores. In this, it's through aplanospores. And other the means are chlamydospores are formed and oodia formation also takes place. And then sexual reproduction is through gametangial copulation rather than gametangial contact, which we have studied earlier in the oomycetes members. So we have to differentiate these characteristics that in oomycetes member you have studied fungal cellulose cell wall, then asexual reproduction to zoospore, sexual reproduction to gametangial contact. In this, we are studying cell wall is of chitin, asexual reproductions to aplanospore, and sexual reproductions to gametangial copulation, which result in the formation of zygospore. The zygospore is the characteristic feature of zygomycetes. The words come from zygospore. So you have to learn this term zygospore in zygomycetes member. Then, if we talk in detail of Rhizopus, the classification belong to kingdom Mycota. All the fungi belong to main kingdom Mycota. Then division Eumycota, subdivision Zygomycotina, class Zygomycetes, order Mucorales, family Mucoraceae, and genus Rhizopus. It's a saprophytic fungi. Some species like Stolonifer is a weak parasite, can cause soft rot in fruit and vegetables. It can cause a fatal disease in humans, that is mucormycosis. In this, lungs and liver and uh, get infected, generally in diabetic patients, or we can say in immunosuppressant patients, in which the disease chances are more. In this, the lungs, livers can get infected with these fungus, right? Now, its vegetative structure, it is characterized by its well-branched cottony appearance, in which you will find a cenocytic hyphae. In this picture also you can see the cottony growth of Rhizopus, right? And then if we study in detail, the mycelium consists of a network of hyphae, and its hyphae is are of three types. The lower hyphae, which are the horizontal hyphae, which are above the substratum, are the stolon. The hyphae which get attached to the substratum are rhizoids, and which get erect, which comes above the soil, or we can say which comes above from the substratum, are erect in form, are spongiophores, and they produce reproductive structures at the terminal and called sporangiospores. In this diagram you can see the stolon, which is in the horizontal form, right? This is a stolon which is running in the horizontal form. Then below that, that is rhizoids, which help in uh, attachment and water absorption from the substratum. And in the erect form there are sporangiophores, on which Sporangium are present in which sporangiospores are formed, means spores are forming at the end part. The spores are aplanospores that are forming in these uh, sporangium, right? Then its cell wall. If we talk of its cell structure, then cell wall is made up of chitin, chitosan is the primary constituents. Then other component can be glucosamine, galactosan, proteins and lipid. Its protoplasm contain nuclei, ribosome, mitochondria, endoplasmic reticulum. All the organelles are present in the protoplasm. Then nucleus is multinucleate conditions present. There are no walls. It's aseptate and aseptate and xenocytic conditions means multiple nucleus are present throughout the nuclear throughout the hyphae, right? Vegetative reproduction. If we talk of its vegetative reproduction, fragmentation take place. If we see in this diagram, then each hyphae is getting fragmented into a small fragments, and each fragment will develop into a new mycelium. So each hyphae can be broken out into a fragments and it can develop into a new mycelium. So it's an vegetative method through which it can reproduce. Then talking of its asexual reproduction, it forms non-motile spores that are aplanospores. It does not contain flagella, right? So through flagella it becomes motile. In the zoospores, in the Oomycetes member we were talking about zoospores, because they were having the flagella. In aplanospores they do not have flagella, so they are non-motile in nature, right? So in favorable condition, the young hyphae grow up and it's become a solan tip, and in this solan tip it differentiate and form sporangioform, and in this sporangioform sporangia develop, and this sporangia differentiated into two part. Lower one it's then central vacuolated sterile part that is called columella, right? Which is an uh, sterile part, and the outer structure is uh, fertile one, which is known as sporangium, or it's in structure where the spores are forming, right? These spores are formed in the outer part. In this outer part the spores are forming, and in these part numerous spores are formed, and on maturity these sporangium dissecs, after dissection the spores come out and these germinate and the cycle will form. So spore formation take place in Rhizopus, right? Then chlamydospore formation also takes place. The mature mycelium becomes septate, right? In this, the septized formation takes place, and protoplasm of each cell form rounded thick wall structure, right? These thick wall structure you can see in this diagram. These thick wall structures are nothing but chlamydospores, right? So chlamydospores and aplanospore formation take place, and asexual reproductions occur in Rhizopus. Mainly aplanospore uh, formation take place in the favorable conditions. Otherwise, if the conditions are harsh, chlamydospores can also be formed. Then, in sexual reproduction, if we talk of sexual reproduction, first I will take you to the diagram. You can see in this two type of strains are formed. This is plus strain and this is minus strain, right? Two type of hyphae are formed. These hyphae form progametangia. These bulge out and form two structures. This is called progametangia, right? And these progametangia come closer and form a structure that's called isogametangia, right? And these isogametangia contain nuclei. This contain male nuclei, and the other one contain the female nuclei, right? The male and female nuclei is contain two type of nuclei, and these two nuclei now fuse, and this form zygospore, right? So in the diagram, in this diagram through this flow chart also you can see the progametangia of plus strain and progametangia of negative strain come closer and form isogametangia, and through gametangial copulation these coming when these isogametangia come closer, and the nuclei are mixing, this type of process called gametangial copulation, right? So through this gametangial copulation zygospores are formed, which is diploid in nature, and a thick wall will be formed, right? It's a diploid structure, and it can remain under the soil, or it can act as a resistant structure. It have thick cell wall, it can enter the dormant stages, store nutrients, and in on the appropriate environment stimuli, whenever it get the environment stimuli like light, temperature, and nutrition, whatever it's required, it will get induced and germinate and grow, right? This characteristic feature you have to remember it for the zygospore. It's in resistant structure, right? And then this zygospore will germinate and form sporangiospore. These sporangiospore will germinate and form spores in the sporangium, and it will again enter into the cycle and form mycelium. In the diagram also you can see these zygospores germinate and form zygosporangium. In this meiospores are formed, and these meiospores germinate and form the plus and minus hyphae, plus and minus strain hyphae again. So you can see these two structures. So I think you have a clear idea of the sexual reproduction, right? Now if we talk of its Rhizopus and Mucor differentiation in the characteristic feature, then Rhizopus is we have already stated that it's differentiated in stolon, rhizoid, and sporangiophore, while in Mucor mycelium is not differentiated into three kinds of IP. It's only one kind of hyphae it's being there, right? Rhizopus, in Rhizopus rhizoids are present, while in Mucor rhizoids are absent. In Rhizopus sporangioforms are formed in trough, while in Mucor sporangiophores are formed, but the hyphae is not differentiated. Sporangioform still are formed, right? But they are formed at and form generally in a single form. They are not formed in a drafted form, but singly they are produced at the end, and the spores are generally disseminated by the winds easily in the rhizopus, while in mucor it is attached to the columella, and it's not easy to disseminate the spores through wind, right? If we talk of its structure, you can see the rhizopus looks like this. In this rhizoids are present, while in mucor the rhizoids are absent, and the sporangioforms are forming in the single form, while in the rhizopus it's formed in the uh, tuft, and they are in the groups, right? So MCQs for competitive examination, species causing mucormycosis. So we have already studied that rhizopus cause this fatal disease in humans. Then mucor will also cause the disease, and lysithemia is also an organism uh, of this mucor family, and it also causes disease. So option will be all of above. Then fungal species commonly known as uh, black bread mold. It is Rhizopus stoloni per, right? Then zygospore formation is a characteristic feature of, then option will be Mucor and Rhizopus. Then metabolic product of some species act as an intermediate in this synthesis of semi-synthetic steroid hormones. Then answer will be Rhizopus. You can see that in 1952, Peterson and Murray developed a process that used the Rhizopus to oxidize protosterone into a compound that was readily converted into cortisone. So it acts as an intermediate in synthesis of semi-synthetic steroid hormone. So these type of questions may arise in the competitive exams. Uh, then if one or two line questions may also arise, that you may write the industrial application of Rhizopus. Then Rhizopus is used in food and feed industries. Indonesian fermented soybean food is being produced uh, by using Rhizopus species, that is tempeh. Then it's used in industries, industrial enzymes preparation, like amylase, pectinase, cellulase, proteinase, right? Then it's also used in uh, synthesize metabolic compounds like glucose, fructose, and sucrose, and also used in waste and wastewater treatment. So I hope you have liked the video. If you have liked the video, share it. If you are new to my channel, please subscribe it. Thank you for watching.